And now, our next presenter is Sebastian Choi. He is a postdoc in the lab of David Ginty at Harvard Medical School. You can find him on Twitter at Sebastian Choi, which we'll put in the chat box. He just had a paper published yesterday. Congratulations. So you can also check out a link to that paper in the chat box below. Sebastian, take it away.
Okay. Thank you for the introduction today. And my name is Seungwon and Sebastian Choi. I'm a postdoc in David Ginty's lab at Harvard Medical School. And today I'll talk to you about the project that I've been working on the past few years, which was focused on the ascending spinal pathways that convey affective touch and pain signals to the brain. Every day, we experience a myriad of somatosensory stimuli, the hugs from loved ones, warm shower, and mosquito bite, and sore muscles after a workout. All these tactile, thermal, and itch and nociceptive signals are first detected by primary sensory neurons, and then propagated into the spinal cord where they're processed. And then these signals are transmitted to the brain through ascending spinal pathways. The primary sensory neuron subtypes that innervate the skin and detect a wide range of somatosensory stimuli have been identified and characterized. So here are examples. There are low threshold mechanoreceptor neurons that detect light touch and proprioceptors and nociceptors and thermoreceptors that detect thermal stimuli and proprioceptors that detect itch stimuli. And in the field, we have the genetic access to almost every single subtype of primary sensory neurons. In contrast, however, very little is known about how these peripheral signals are integrated and processed within the spinal cord, and how these signals are conveyed to the brain by a spinal cord projection neuron to generate our somatosensory perception and behavior responses. So the goal of my study was to better understand the circuit logic of these ascending spinal pathways. And I've been focused on this anterolateral pathway, a major ascending spinal pathway that is known to convey mainly the temperature and pain information from the periphery to the brain. And these neurons receive nociceptor inputs, and their axons cross the midline in the spinal cord and innervate the contralateral side of the brain. And I was particularly interested in this one region, lateral parabrachial nucleus, because this region receives the most dense innervation from the spinal cord. And this ascending pathway is termed spinal parabrachial or SPB pathway. And again, as I said, we have in the field genetic access to all these, most of the primary sensory neuron subtypes. In contrast, we almost completely lack the mouse genetic tools that label different subtypes of projection neurons in the spinal cord. So we started this project by generating mouse genetic tools to label different subtypes of projection neurons in the anterolateral pathway. And we generated first the Tacr1 positive tool because Tacr1 positive neurons were the only known population that label a major subset of SPB neurons. And then we also took an unbiased screening approach to identify unknown and novel subtypes of projection neurons. So we conducted in silico screening of over 1,300 the back GFP transgenic mouse lines from the genset. And we identified this one line, Gpr83 GFP, labeled a major subset of SPB neurons. So these neurons are mainly located in the superficial lamina of the spinal cord dorsal horn, where these, the projection neurons are enriched. And with the retrograde labeling experiments, I was able to show that these neurons label a major subset of SPB neurons. And with the mouse genetic tools that we developed, we were able to visualize both of these populations at the same time. So here in these images, TD tomato neurons represent the Tacr1 positive projection neurons, and green neurons represent Gpr83 neurons. So as you can see here, these two neurons are largely non-overlapping populations. And then we looked at the brain target innervation of these two SPB subtypes. So interestingly, it turns out that the Tacr1 positive neurons mainly form synaptic terminals in these two subnuclei of the PBN, whereas Gpr83 projection neurons uniquely innervate DL and EL subnuclei in addition to the IL subnucleus. These results suggested these two SPB neurons innervate the PBN in a zonally segregated manner. So now we have these two anatomically distinct subtypes of SPB neurons. So next we wanted to investigate the function of these two projection neurons, particularly what somatosensory stimuli or signals they convey to the brain. So before I go into the detail of the data, I wanted to give you some background. So textbook view of ascending touch pathway mainly emphasizes the dorsal column medial lemniscal pathway, where the touch signals are first detected by mechanosensory neurons, and these signals are transmitted to the spinal cord and brainstem and to the thalamus, and then somatosensory cortex. So this is a classical touch pathway. In contrast, as I said earlier in the introduction, anterolateral pathway, mainly known to convey the pain and temperature information to the brain. However, we think that this newly identified Gpr83 positive SPB pathway form dedicated and unique touch pathway within the anterolateral tract that convey tactile signals from the spinal cord to the lateral parabrachial nucleus. And the PBN in turn broadcasts these tactile signals to other regions of the brain, including the amygdala and hypothalamus. So I'll walk through some of the data that support this idea. So we first wanted to look at the physiological response properties of these two projection neurons. So we collaborated with Sarah Ross and Rick Corber's lab, and they use this ex vivo skin spinal cord preparation. And Junichi, the postdoc at the time, performed wholesale patch clamp recordings on these targeted projection neurons while giving different types of stimuli onto the skin. So he measured the change in the firing rate upon the application of different types of somatosensory stimuli. And one of the most unique properties of Gpr83 neurons was that compared to the Tacr1 projection neuron, they're highly sensitive to mechanical stimuli. So here in this summary rate of plot, the left panel is a percentage of responders and right panel is the firing rate. As you can see here, the Gpr83 projection neurons are they're very responsive to the wide range of foam-fry filament skin indentation. Now, when they respond with much more higher firing rate. And whereas the Tacr1 projection neurons are more responsive to thermal stimuli, especially in the innocuous range. Here's a summary plot. So to define the spinal cord circuit mechanism underlying this mechanosensitive response properties of Gpr83 projection neurons, we performed the channelopsin assisted circuit mapping experiment using acute spinal cord splice preparation, and also this complicated mouse genetic strategy that allowed us to specifically stimulate different types of sensory neurons while recording from specific types of the projection neuron in the spinal cord. So axonal terminal stimulation of mechanosensory neurons, including the MRGB4 positive non-peptidergic neurons and TRAC-B positive a delta low threshold mechanoreceptor neurons, evoked large light activity currents and also action potential firing in Gpr83 projection neurons, but not in the Tacr1 neurons. So these suggested Gpr83 neurons receive strong synaptic inputs from the mechanosensory neurons, and this can explain its mechanosensitive response properties. And then we turn to the anatomy of the spinal parabrachial pathway. What is really interesting about this pathway is that they have a unique organizational property. So in this experiment, I injected a cre-dependent alkaline phosphatase virus unilaterally into the right-hand side of the spinal cord and performed tissue clearing and the Holman AP staining to visualize ascending axonal projection to the brain. And as I said earlier, the ascending pathways within the anterolateral tract, they mainly innervate the contralateral side of the brain, for example, here in the midbrain PAG or superior colliculus. However, the PBN receives predominantly bilateral inputs from the spinal cord. Another interesting anatomical feature of this pathway is that they lack somatotopic organization. So in this experiment, I injected three different colors of synaptophysin viruses into cervical, thoracic, and lumbar spinal cord. and I examined their synaptic terminal distribution within the PBN. And surprisingly, these three different colors of synaptic terminals were intermingled within the PBN, suggesting that there's no clear somatotopic organization in this spinal parabrachial pathway. So these unique anatomical properties led us to suspect that maybe this pathway convey affective component of somatosensation rather than discriminative component of somatosensation. To directly test this idea, we performed this optogenetics coupled instrumental conditioning assay where animals receive selective optogenetic stimulation of either of these SPB pathway when they press the active lever but not inactive lever. So we tested a different optical strength, and we started with the high power, strong activation of either of these pathway and then dialed down. And down to the 1 milliwatt strong activation of either Gpr83 or Tacr1 pathway reduced the lever presses over time, suggesting that a strong activation of these pathways is aversive to the animals. And surprisingly, when we stimulated the Gpr83 with the low intensity light stimulation, it promoted positive reinforcement. In other words, animals increased the lever presses over time, suggesting that low intensity stimulation of Gpr83 pathway is appetitive to animals. So these results suggest that this Gpr83 pathway can be associated with either positive or negative valence in a scalable and stimulus intensity dependent manner. To summarize, I didn't have much time to show you this part today, but we showed that these two pathways form parallel ascending circuit modules that cooperate to convey nociceptive signals to the brain. And as I described today's talk, this newly identified Gpr83 pathway constitutes a mechanosensory limb of the anterolateral pathway that conveys an affective component of touch. So I showed you these neurons are highly sensitive to mechanical stimuli. They receive strong synaptic inputs from the mechanosensory neurons, and they form bilateral and non-somatotopic organized synaptic inputs into the PBN. And they can induce the behaviors associated with either positive or negative valence in a stimulus-dependent manner. So overall, our study reveals these two anatomically and physiologically and functionally distinct spinal parabrachial pathway that underlie affective touch and pain. So with that, I'd like to thank David, his support, and all the people from David Gintis Lab who contributed to this work, and a collaborator, and the mouse lines and the viruses and the funding source. And with that, I'll take any questions.
Thank you, Sebastian. Very nice talk. We have a question from Asaf. In intact animals, is it rare to record responses of PB neurons to tactile, non-noxious stimuli? Can you comment how this relates to your findings?
So that is a good question. So I think one of the issues that mostly in the field, people have done recordings in the PBN, anesthetized animal. So I think one possibility is that maybe anesthesia can affect the activity of these neurons. And also, as my work sort of suggested that probably depending on the stimulus intensity, it'll recruit. So if you increase the intensity of the mechanical stimulation, it will recruit more and more neurons activated by mechanical stimuli. So I think one might want to have really sensitive physiological measurement to really detect the units that are responsive to the innocuous mechanical stimuli. But at least in our ex vivo preparation, we see pretty strong response to the mechanical stimuli.
We have another question from Rebecca Bohm. Great talk. Is the SPB pathway in mouse comparable to that in human? Do you believe we can expect this pathway to perform the same function in humans?
Oh yeah, that is a really good question. Actually, in humans, this spinal parabrachial pathway has not been much studied. People mostly focused on spinal thalamic pathway that goes to the signal goes to the somatosensory cortex. But there are there are a few the human studies. Actually, one of the some of the human patient studies suggested actually this anterolateral pathway can convey both negative valence associated with the pain signals, and also, but also the gentle touch, or pleasurable touch information associated with the positive valence associated with the gentle touch. Because those studies, what they showed was, for example, the patients with the terminal cancer, they have really extreme pain. So one of the strategies to reduce the pain in those patients was to transact the extending the anterolateral pathway. So basically cutting the anterolateral pathway. And these patients have reduced the pain sensation, but also oftentimes they also show they lose the kind of pleasant feeling, the present feelings associated with the gentle touch, while maintaining the ability to locate or identify the source of the sensory stimuli. So I think those human studies kind of suggested maybe in humans also have conserved roles in terms of conveying this affective component of somatosensory stimuli.
Thanks, Sebastian, again.